My name is Heather Richard. I'm a University of Maine graduate student in the Department of Ecology and Environmental Sciences, working with the Maine-eDNA program. Marshes are really important for absorbing pollution as a nursery for a lot of different types of wildlife, and they're also really important places that can store large amounts of carbon per square foot. I'm looking at the ways that humans impact marsh ecosystem function, and specifically how they impact carbon sequestration by building bridges and roads across tidal creeks. I'm interested in studying marshes and the way that humans impact the way marshes function, specifically the way that they store carbon in their soils. When we build bridges and roads across tidal creeks, we change the upstream habitat, we change the microbial community, and that changes a lot of the ways that carbon functions in the system. I'm interested in looking at how marsh restrictions change the way marshes function upstream as compared to downstream. Environmental DNA, or eDNA, is a great tool to use for looking at microbial communities because it's very difficult to study these small organisms in the lab and outside of the field. You'd need to identify them based on how they look under the microscope, which is really difficult and impossible in some cases. And then we can't grow many of these microbes in the lab because they just don't live in that environment. So by capturing the DNA that's found within the soil and sequencing that DNA, we can understand all the different species that are living together and possibly contributing to the ecosystem function of the marsh. So we identify marshes that have restrictions and some that don't, so we have a comparison, and we sample along the creek at specific points in space, equidistant apart. And what we'll do is when we get the information back on which species are living where, we can compare those upstream versus downstream. We expect that there'd be a difference because there's a salinity gradient and the vegetation might be different upstream versus downstream, but we can look at comparisons for how the rate of change along the stream might be different based on whether or not a restriction is present. Microbes like bacteria and fungi are really important because they could be pathogenic or they could be beneficial microbes that help the plant's function, and they all together contribute towards the way the ecosystem functions as a whole. The plants that live there might be either harmed or enhanced by the amount of the types of microbes that live there. But also the microbes are really important for whether or not they're eating carbon and releasing carbon dioxide, or whether or not they're releasing methane. And different species or different groups of microbes might have different functions that we can imply based on who's living where. My hope for this work is that we know marshes better by the end of it, and we can make better predictions for all the variation that might be occurring in terms of our ecosystem function, the way that bridges and roads change what's happening, and the way that we model the amount of carbon that's stored and the carbon that could potentially be stored within each marsh. It's really important as we're working with climate change and trying to be climate neutral, it's really important that we have an accurate assessment of the net amount of carbon that's being stored in the environment versus what's being released. And marshes release not only carbon dioxide, but they can also release methane, which is a potent greenhouse gas. And sometimes bridges and roads can create environments where bacteria love producing methane. So by studying how restrictions impact marsh environments and change the communities, we might be able to predict more what is being released and what is being stored within each marsh environment.